So muscles perform various vital functions in body. They perform body movements like walking, manipulating objects, keeping objects, holding objects, and then putting on another place. They help in body positioning. For example, there are muscles that keep our body in erect position, uh, and there are muscles along the blood vessels and other tubes of the body to keep them open. These muscles are they have a continuous tone to keep the tubes open. So they also help in storing and moving substances. For example, they store like calcium, glycogen, and proteins. So then they also help in generating heat, like in shivering. They produce heat that is used to warm up the bodies. Similarly, in other movements of the muscle, there is production of heat as a byproduct. So it also the muscles they also help in communication like speaking, writing, gesturing. They also help in respiration. The movements of ribs and diaphragm is dependent on those muscles, and then constriction of organs and vessels. They help to keep the like I told previously that they keep the shape of the organs and vessels proper. They help in heartbeat. The cardiac muscle they help in heartbeat. So what is the function of a muscle? A function of a muscle is only contraction, not uh, relaxation, because the muscles they contract actively by using energy and they get relaxed passively by uh, antagonistic muscle, by force of gravity, or by the fluid pressure in the tubes. For example, our bicep and tricep they get relaxed by contraction of their counterpart, and the muscles in uh, blood vessel they get relaxed when there is fluid pressure. So the contraction is the main function of the muscle, it is not relaxation. So there are four other properties of a muscle, that is contractility, excitability, extensibility, and elasticity. Contractility is that a muscle can contract, it can get shortened in its length, and excitability is the characteristic by which a muscle can be excited. If a nerve impulse come on a muscle, it can get excited, mean action potential can run on a muscle. And extensibility mean the muscles they have property that they can be extended beyond their normal length. Elasticity is actually the elastic property of the muscles, mean we can stretch muscle beyond their length and then they can recoil like an elastic rubber band. So the muscles they have four properties: contractility, excitability, extensibility, and elasticity. Types and function of the muscles. You know very well there are three types of muscles, that are skeletal muscle, smooth muscle, and cardiac muscle. And their location, cell shape, number of nuclei, shape of the placement of nuclei, and their cell to cell interaction, striation, control, capability of spontaneous contraction, and functions they differ a lot. For example, in skeletal muscles, we know they are attached to bones, they are attached to skeletal system, and their cells are very long and cylindrical. They have multiple nuclei, and these nuclei they are located on the periphery of the cell membrane, because inside of the cells the myofibril are filled that are helping in contraction, that is the actin and myosin. And then these cells, they are independent cell, mean they don't have interconnection, they are not connected to each other, they are independent cell, they are only controlled by the nerves. So the striation on these muscles, skeletal muscles, is present because of the difference in thickness of the myofibrils thickness and the actin and myosin, and the overlap of actin and myosin. So we will study them later on. These muscles, they are controlled most of the time. Their control is voluntary. It means with the will of the individual, and sometimes they are involuntary in the form of reflexes. For example, the reflexes, they are uh, they involve only few intermediate neuron or even no intermediate neuron. It means the sensation is carried by a sensory neuron from the sensory organ to the central nervous system, that is either spinal cord or the uh, medulla oblongata in brain. And then the intermediate neuron, if it is present, then there are few, and if they are not present, then this sensory neuron, it directly sends signal to the motor neuron to control the contraction of that muscle. So the skeletal muscle, they have their reflexes mostly in spinal cord, and these muscles, they are not capable of spontaneous contraction. Their function is to move the body and for locomotion or for the body movements, that is the movements of the organs. So the smooth muscles, they are located in the walls of hollow organs, like in blood vessels, in the tubes of the digestive system, reproductive system, respiratory system, and they are also present in eyes, glands, and skin. Their cells, they are spindle shaped, and these are shorter cells than the skeletal muscles. And these cells, they have a single nucleus per cell, and these nuclei, they are centrally located inside the uh, smooth muscle cell. And these cells, they are interconnected to each other, mean they have bridges between them, they can exchange material to each other. So if we excite one cell, then it can excite the other cells in connection. So these cells, they don't have striation because the smooth muscles, they don't have the sarcomere like in skeletal muscle. In skeletal muscles, the actin, they are present, they are making a cylindrical shape structure, and myosin is sliding in that cylinder. But in case of smooth muscles, the actin and myosin, they are not like that. The actin are less in number, and they are like the actin and myosin, they have connection like they are running on each other. And then the actin, they are not enclosing entirely the uh, myosin. These smooth muscles, they are involuntary, mean we cannot control, or an individual cannot control them. They are controlled by higher brain center, and the control is in medulla oblongata. So these muscles, they have capacity to contract spontaneously, and then their function are like the location is showing their function. For example, their uh, muscle that are present in blood vessels are helping in the regulation of blood vessels, transport of blood, and then the emptying of the urinary bladder. They are helping in changing pupil size, contraction of gland ducts, movements of hair, and so many other functions. The cardiac muscle, they are present in heart. These are cylindrical like skeletal muscle, but they are shorter in length, and then uh, they are branched, not like skeletal muscle. Skeletal muscle are cylindrical, but they are unbranched. Cardiac muscle are branched, mean they have many uh, branches, and then their uh, nucleus per cell is only one, and they are also centrally located. These cardiac muscle, they have special type of connection between cells. These are called as intercalated discs. Intercalated discs are actually the merger of two types of connections. These cells, they are connected through gap junction. It means they can exchange material, as well as they have the desmosomes between them. Desmosomes are protein structure that keeps the two cells stitched together. So these stitching material is stitching the membrane of two cells, and the gap junction are helping to move material from one cell to another. So again, like skeletal smooth muscle, the cardiac muscle, if one cell get excited, it can excite another cell by transporting material. So the striation on cardiac muscle is present because the cardiac muscle, they have same sarcomere like in skeletal muscles. The cardiac muscles, they are involuntary, mean they are not under control of an individual. They are controlled, they are most of the time, the cardiac muscles, they can work themselves without any input from the higher brain center. It means if we keep them outside of the body and give all the requirements to them, they can work on them on themselves. Mean they can work spontaneously. They don't need even input from the brain, the involuntary control. So the involuntary control is just to keep them in their homeostatic condition, their optimal work. The involuntary control does not allow the cardiac muscle to work beyond their requirement. So then these muscles, they are capable of spontaneous contraction, because we know there are SA node and AV node, sinoatrial node, and the atrioventricular node. These are the muscle cells, cardiac muscle cells, that are uh, um, modified to produce action potential by themselves, so they are capable of spontaneous contraction. uh, the cardiac muscles, they are pumping blood, and they are producing force to push the blood into the blood vessel. The cardiac muscles are involuntary, like smooth muscles. They are being controlled from higher brain center, and most of the time they even don't need any input from higher brain center. Mean they are independent. They can contract themselves. They don't need any instruction most of the time, but in special cases they need input to maintain homeostasis, mean to work in optimal to work for performing optimal functions. So the higher brain center is just regulating them. Otherwise they are capable of contracting themselves. The cardiac muscles, they have the only function to push blood into the blood vessels for transportation of the materials. So the structure of skeletal muscles. Skeletal muscles are composed of muscle fibers, that is the muscle cells. So muscle cells in skeletal muscle, in smooth muscle, in cardiac muscle, they are called as muscle fibers. So a complete cell is a muscle fiber. So the skeletal muscles, they are made up of muscle fibers. They have connective tissue. Here it is written as CT. it is connective tissue. Then they have blood vessels and nerves in them. So there are mainly four components of a muscle fibers or cells, connective tissue, blood vessels and nerves. So the first, the cells. What are the cells of the skeletal muscle and how they are originated? The origin of the muscle cells is from myoblasts. These myoblasts, they are undifferentiated cells. They originate from, as we know from previous lecture, the mesoderm. In gastrudation, we have three layers, germs layer, the ectoderm, mesoderm and endoderm. The mesoderm, the internal, the inner layer, it give rise to the connective tissue and muscles. So the muscle, the myoblasts, these are the undifferentiated cells. They originate from that mesoderm. These cells, they are multinucleated, and so the myoblasts, they uh, divide their nuclei during development, but the cytoplasm, they are not divided, and they make a syncytium, so they become multinucleated, and few of these myoblast cells, they get differentiated into muscle cells, and other remain as undifferentiated cells. We can see here them as satellite cells. So these satellite cells, they are present even in mature cells, in mature muscles, and they may help in the repairing of the muscles. So we know the number of the muscle cells is same as we got on our birth, and the only the size of the muscle is increased. So the muscles, they can store more protein and they become hypertrophied, mean their size is increased. So in the adults, it is only the increase in size of muscle but not the number of muscle cells. So the size of muscle cells is increased and the size of the whole muscle is increased. In case of few diseases are even lacking the nutrients and the uh, lacking exercise, the muscles, they can get reduced in size, that is called as hypertrophy. So the muscles, their size is quite flexible, mean they can increase their cell size, they can decrease their cell size. If they increase cell size, that is called as hypertrophied, and if they decrease cell size, then it is called as the hypertrophy. So the number is fixed. Skeletal muscle cells, they have striation on them, mean they have the sarcomeres, and because of these sarcomeres, the arrangement of actin and myosin, under light microscope, they appear to have bands of light and dark band. We'll see them later on. The skeletal muscles, they have three main layers of connective tissues. These layers of connective tissues are called as fascia. Fascia mean a detachable layer, and like the protectors on our mobile phone. These three layers of connective tissues on skeletal muscles are from outer to inner is epimysium, perimysium and endomysium. So these are the three main layers. The endomysium, it encloses a muscle cell. The perimysium, it encloses a group of cells that is called as fascicle. A group of muscle cells in skeletal muscle is called as fascicle. And epimysium is enclosing the entire muscle. It means enclosing many fasciculi, many groups of muscle cells. So the epimysium, it is the thicker most layer because it is covering the muscles from outside. And then perimysium, it is less thicker than epimysium, but it is more thicker than endomysium. And if we see structure of a muscle, skeletal muscle, The innermost material that is of importance are the myofilaments. The myofilaments, there are two main myofilaments, that is the actin and myosin. These are the actin and myosin protein, contractile protein. Their arrangement makes myofibril. These myofibrils, they are fibers that are contractile. They are made up of actin and myosin. And then these myofibrils, they are present, they are packed in muscle cells. The muscle cells, they are called as muscle fibers. The muscle fibers, they are called muscle fiber because they are elongated in length. So the muscle fibers, they are like other cells. They have same types of composition, mean they have all the organelles like mitochondria, and then they have nuclei, and then they are having the plasma membrane, that is also called as sarcolemma, that is with relation to muscle. So it is a layer that is the plasma membrane of the muscle cells. Then these muscle cells, they are, each muscle cell is covered by a connective tissue that run along the whole length of the skeletal muscle cell. That tissue is called as endomysium, that connective tissue that is covering the muscle cell on its and present on its cell membrane. That layers of connective tissue is called as endomysium. So this endomysium is least thicker connective tissue of the three layers. So this endomysium, then it is running along the whole length of the muscle cell, and most of the time it is very wide to the skeletal muscle membrane, and then cell membrane. So the cells that are enclosed by endomysium, these cells, they make groups. These groups, they are called as fascicle, and each of the fascicle is enclosed by a thicker membrane that is called as perimysium. So the fasciculi, they are then enclosed by a more thicker membrane, and that membrane is called as epimysium. Epi mean that is on top of. So these three layers of connective tissues, they are providing certain important function. First of all, we know the connective tissues, they are connecting different types of tissues or same type of tissue. So the connective tissue here is binding the muscle cell to make groups that are called fascicle, and then binding fasciculi to make the whole muscle. So one of the function is binding, as the name depicts, the connective tissue they are connecting, and then they also help to connect the muscle, because these endomysium, perimysium and epimysium, they are running along the whole length of the muscle, and then they get merged into the tendons. Tendons are the connective tissues that are connecting muscles to the bones. So these connective tissues, endomysium, perimysium and epimysium, they are also connecting muscles to the bones. They are, these connective tissues, they are also helping to run the blood vessels and the nerves into the muscles. These, they get connection from the bones, the blood vessels from bones, they come into the muscles, and through these connective tissue, and these blood vessels and the nerves, they go up to the level of each cell. It means they pass through, they come from tendon, they go into the uh, epimysium, they penetrate into the perimysium, and then they reach up to the level of the uh, endomysium, and the blood vessel, they remain outside of the endomysium, but the nerves, they go deep into the uh, onto each and every cell, onto their cell membrane, in order to excite them, in order to give them instruction, because the muscles, they just have to contract under the instructions of nervous system. So the instructions, they are coming through these nerves onto each and every cell of the skeletal muscle, because each and every cell needs instruction because they are independent cells. The skeletal muscle cells, they are not interconnected, so each and every cell is needing that instruction that is coming in the form of these nerves from bone, from the epimysium into the perimysium into the endomysium, and then on to each and every cell. The arteries and veins, they are coming from bones, they are going into the epimysium, they are passing through epimysium, going to the perimysium, and from perimysium they are going up to the level of endomysium, but they remain outside of the endomysium. They are helping to provide oxygen and nutrients to the muscle for contraction and taking waste material back from these muscles. The component of a myofibril or muscle cell are similar to a typical cell. They have sarcolemma, the cell membrane, sarcomere, flesh or the muscles, and lemma means sheet or a layer. They have cytoplasm, that is also called as sarcoplasm. The cytoplasm of the muscle cell has a protein that is not present in other cells. That protein is called as myoglobin. So myoglobin is a counterpart of hemoglobin. It is only present in muscle cells. Myoglobin and hemoglobin have few similarities. Both are helping to carry oxygen. Both are having globulin chain, and then the iron element for their carrying oxygen. So myoglobin and hemoglobin, they are different in structure and function. uh, these differences, for example, hemoglobin, we know it is present in RBCs. Myoglobin is present only in muscle cells. So the hemoglobin in case of human being, it is present in blood, mean it is inside RBCs. Myoglobin, when it is present in blood, it means the muscle cell are damaged, so the myoglobin is leaked into the blood. So myoglobin is usually, in normal conditions, it is only inside the muscle cells. The second difference is in their structure. The hemoglobin is tetrameric. It is having the four chains, while the myoglobin, it is monomeric, mean it has a single polypeptide chain. So the hemoglobin can get bind to, because it has four chains, four heme groups that are containing iron, and these four heme groups, they can carry four molecules at a time, molecules of oxygen at a time. So a single hemoglobin molecule can carry four oxygens, but a myoglobin, because it is monomeric, so it can contain only one molecule of myoglobin can carry one molecule of oxygen at a time. So then the hemoglobin, it has affinity for carbon dioxide, the protons and nitric oxide, but myoglobin has affinity for only oxygen. Myoglobin can firmly bind to oxygen than the hemoglobin. So myoglobin, it helps the muscle cells to uh, keep the oxygen stored. Hemoglobin is mostly for the transportation of oxygen, but myoglobin is for the storage of oxygen in muscle cells. So the myoglobin, it gets oxygen from outside of the cell, from extracellular material, and then it carry it to the mitochondria. So the then another structure that is called as T-tubule or transverse tubules. These are the invagination of the plasma membrane of the or sarcolemma of the muscle cells. These invaginations, they are at right angle to the length of the muscle cell, so they are called as T-tubules. Now we'll see them later. Then the muscle cells, skeletal muscle cell, they have multiple nuclei, they have multiple mitochondria. These mitochondria, they are arranged along T tubules or transverse tubules, the needle-like transverse tubule that are going deep into the muscle cell. And then the muscle cells, they have myofibrils in the form of myofilaments that are made up of actin and myosin proteins. These are the contractile protein that slide on each other to produce contraction. Here in this diagram we are looking at a muscle cell. So this muscle cell is having uh, nuclei. This muscle cell is bound with plasma membrane or sarcolemma, and then we can see prominent nuclei that are present on the periphery, and we can see capillaries that are reaching up to the level of endomysium, and then inside we can see there are pores, a little dots you can see on the plasma lemma. These dots are actually transverse tubules. These are the needle-like tubules, blind-ended tubules that are going deep into the length of the muscle. So here in the section we can see the transverse tubules, they are going deep into the cell, so they are running along the myofibrils. So myofibril, here we can see mitochondria, they are also present on the periphery along the length of these T tubules. So then two mitochondria, they are present on each side of a T tubule. So this whole structure is called as triad. We may see them later on. So a T tubule and two mitochondria on its periphery, this whole structure is called as a triad, mean a structure of three components, a T tubule and the uh, mitochondria. So these T tubules, because they are originated from plasma membrane, so they have all the characteristics of the plasma membrane, mean they are semi-permeable like plasma membranes. They have channels and everything. So the uh, myofibrils, they are made up of myofilaments. We can see here actin myofilament, that are the thinner filaments, and then myosin myofilaments, they are thicker proteins. So the arrangement is here, we can see their arrangement, and this arrangement is called as sarcomere. In case of skeletal muscles we can see a two-dimensional structure, but in actually in three-dimensional structure, the actin, they make a tube-like structure. So the tubes of actin, they have myosin running in them. So we'll see them later. As we know, the uh, myofibrils, they are made up of two types of myofilaments, actin and myosin myofilaments. Actin are the thinner myofilaments and the myosin are thicker filaments. Actin is not a single protein. It is made up of three main types of proteins. These three types of proteins are G-actin, or it is also called as fibrous actin, or it is globular actin, and the second protein is tropomyosin, that is a fibrous protein, thread-like, rope-like structure, and then a smaller protein that is called as troponin. Troponin is having three components. So we start from G-actin or globular actin, or it is called fibrous actin. It is made up of globules. So the monomers, they are globular, and these globular monomers, about 200 globular monomers, they make a single molecule of G-actin. So these globules, they are present in the form of two fibers, and these fibers, they are twisted like a helix, and that is called as an actin helix. These globules of G-actin, they have binding sites for myosin. we can see here the black dots, they are actually the active sites for myosin. Myosin has to bind here. So the tropomyosin, it is made up of two threads of protein that are interwoven like a rope-like structure, and a single molecule of tropomyosin reside on the 7 globules of G-actin. So tropomyosin is actually covering active sites for myosin that are present on G-actin. So a single tropomyosin molecule can cover 7 G-actin molecules. Then the uh, troponin, it is made up of three components called as C-troponin, I-troponin, and it is T-troponin. The C-troponin has to bind with calcium. The I-troponin has to bind with actin or G-actin, and T-troponin has to bind with tropomyosin. Mean three components are binding with three different things. One component is binding with calcium, another component is binding with tropomyosin, and another component is binding with actin. So these troponin, they are present only in the grooves of these helical structure that are made from G-actin and tropomyosin. Myosin is a thicker protein, and a single molecule is made up of two heavy chain and two light chain. Here in this diagram we can see that a single molecule is made up of two heavy chains. These heavy chains, they have tails and they have heads. So these tails, they are twisted in the form of a rope, and the heads, they are free. And in next diagram we can see the heads, they have attack with uh, light chains, and these light chains, they have active sites for binding with actin to make cross bridges with actin, and they have ATPase site. It means ATP get bind to this site and get hydrolyzed, and energy is stored in the myosin. So myosin get energized when they hydrolyze ATPs. So this region of the heavy chain that is connecting head to tail, this region is called as hinge region. Like a hinge joint, it can get straightened and it can bend. When it is energized, it can bend, and when it is not energized, it can get straightened. A single in a sarcomere, there are about 300 myosin molecules, and these 300 molecules, here we are looking in the diagram B. These 300 molecules, they are arranged in left and right side, and their tails are attached to each other at a central point through a protein that is attached, linker protein. So the 150 molecules are attacked they are oriented toward left, and 150 are oriented toward right side. So a single sarcomere is having 300 molecules, 150 arranged toward left and 150 toward right in opposite directions. There are two other important proteins that are nibulin and titin. Nibulin and titin are actually cytoskeleton of muscle cells, and the nibulin, it like cytoskeleton in other cells, their function is to support the uh, muscular structures like a skeleton does. So nibulin is providing support to actin. It keeps its structure and shape and position maintained. Similarly, the titin It is the cytoskeleton protein that is supporting myosin and maintaining its function, its structure, its shape, its position. So these are the two important cytoskeletal proteins. The arrangement of myosin and actin in a myofibril is called as sarcomere. So sarcomere is also called as a functional unit of a muscle. The sarcomere may be long enough to reach the whole length of a muscle cell. In case of shorter muscle cells, a single sarcomere can be present. But in case of longer muscles, the sarcomere, they are arranged end to end or back to back and reach to the whole muscle length. The sarcomere, they have because they have overlap of actin and myosin, they have attachment of actin to actin, they have attachment of myosin to myosin, so they give rise to few characteristics. We'll talk about them later. These characteristics are Z disc, that is the attachment of actin of two different sarcomere, uh, and then M line, that is the attachment of myosin, the A band, that is the overlap of actin and myosin, and I band, that is called as the only actin fiber that does not overlap on myosin. In this diagram we can see the arrangement of actin and myosin in a sarcomere. The uppermost diagram shows that the sarcomere, they are arranged end to end with each other, and it is also showing two dimensional. In front it is two dimensional arrangement of the actin and myosin. We can see the blue color line, they are actin, and the inner thicker layer is the myosin. So from two dimensions we can see there are two actin that are overlapping a myosin. Actually, in a three-dimensional diagram, in diagram B, the cross section, we can see there are six dots, smaller dots, that are enclosing larger dots or thicker dot. These six dots, they are actin. So the actin dots, the actin molecules, six actin molecules, they are enclosing a myosin molecule. It means six actin myofilaments, they are making a tube-like structure, and myosin is sliding in them. So in three-dimensional structure, it is the six actin molecule myofilaments that are enclosing a single myosin myofilament. So same is in A diagram, that is the microscopic diagram. Here in A diagram we are looking at a myofibril. This myofibril has in center from one Z disc to another Z disc a complete sarcomere. So this sarcomere is lined with Z discs. Z discs are actually the point of attachment of actin from two different sarcomere. So this point of attachment is thicker than the rest of actin. So under microscope it makes a Z disc or Z line. It is a darker region than the other actin region. Then we have a central line, that is the M line. It is in the center of sarcomere, a thicker line. This line is present on the myosin. The myosin molecule that were arranged in, we saw there are about 300 myosin myofilaments in a myosin molecule in a sarcomere that are arranged in opposite direction. So 150 are arranged on left side and 150 on right side. So the tails of these myosin molecule, they are attached to each other at a region a line that is called as M line because there is a linker protein, so this region is also thicker than myosin, so it appears to be dark than rest of the myosin, so it is a distinct region that is called as a M line. In B diagram we can see in detail the Z disc. These are the linker protein or the thicker protein, and then the M line, it is again a linker protein that is attaching the myofilaments, myosin myofilament that are arranged in opposite direction. So why the Z disc is thicker? Because here the actin filaments from opposite are back to back arranged sarcomere, they get merged, and then there is a linker protein that is uh, helping to connect these actin myofilaments from two sarcomere. This linker protein is called as actinin. Nibulin and titin, they also get merged into this region, so this region Z disc is thicker than the rest of the actin. So we can say Z disc is made up of actinin, the linker protein, the actin from the back-to-back arranged sarcomere, nibulin and titin. The myosin are linked at M line. So M line is the darker line because it is made up of myomycin, thick myosin, that is the linker protein that is linking the oppositely arranged myosin myofilament molecules. It is having creatine kinase that is helping to uh, hydrolyze the creatine phosphate. So we'll see it later. Here is a microscopic structure of sarcomere. We can see sarcomere is start from a Z disc to another Z disc. The Z discs, as we saw previously, they are thicker. They are made up of four types of proteins. That is the linker protein, alpha-actinin, the nibulin, titin, and the actin. And so it is thicker than the actin. And then we have a central line that is called as M line. M line is actually the attachment of myosin molecules on opposite direction. So M line is made up of, we said it is a special type of myosin linker protein, and then it is the creatine kinase. So the rest we are looking at a H zone. H zone is actually Heller zone. It is in center where there is only myosin. It is not overlap to the actin. So you can go back to the previous diagram and see the H zone is only including myosin. So myosin is thicker, but it becomes more thick when it is overlapped by actin. So where there is in central region where there is no overlapping between myosin and actin and it is only myosin, this is brighter region, so it is called as Heller zone or the brighter zone. So then we have bands, the region that is including myosin, either it is overlapped by actin or not. This region is called as A band because it is anisotropic. Light cannot cross this region, so it appears to be darker in under microscope. So this is called as anisotropic. The light cannot pass. This is the whole region where there is myosin. In I band it is only actin. So the I band is isotropic band. The light can pass through this region because actin they are thin, so light can easily cross. So under microscope is this region appears to be brighter. Why they are called Z disc, M line, H zone, I A band? Because Z disc is from German word Zwischenscheibe, that is the disc in between the I band. M line is the middle Scheibe, the middle line. H zone is heller zone, the brighter zone. I band isotropic, A band anisotropic. Here we can see in I band it is only actin, and in A band it is myosin and actin that are overlapped or not. So when the muscle contract, these myosin they slide into actin, and so the while contraction I band get shorter but A band remain fixed. So A band is always fixed. It is the I band that get larger when the muscle relaxes. The myosin slide out of the actin, and it gets shorter when the myosin slide into the actin tubes.